Today's episode is sponsored by Science.bio Premier Nootropics. You can find the link in the description. We're looking at a study titled Seletracetam Enhances Short-Term Depression in Vitro, published in Epilepsy Research in November 2015. This study investigates how Seletracetam, an analog of the anti-epileptic drug Seletracetam, affects synaptic transmission in the hippocampus. Could you explain what Seletracetam and Seletracetam are and how they relate to epilepsy?
Seletracetam, or SEL, and Seletracetam LEV are both anti-epileptic drugs. They're designed to prevent seizures in people with epilepsy. They belong to a class of drugs known as pyrrolidinones.
Interesting. This study found that SEL decreases those later EPSPs in long trains of stimuli. Can you explain what EPSPs are and how their reduction might relate to seizure suppression?
EPSPs, or excitatory postsynaptic potentials, are electrical signals that neurons use to communicate. When a neuron receives enough excitatory signals, it fires. Sending a signal to other neurons, a rapid-fire chain of these signals can lead to a seizure. By reducing the strength of those later EPSPs, SEL could help to prevent seizures. It's like SEL is kind of lowering the volume on those signals.
The study also found that SEL's effect depends on accessing the inside of synaptic vesicles and binding to a protein called SV2A. Can you expand on that for our listener?
Synaptic vesicles are tiny sacs within neurons that hold neurotransmitters, the chemical messengers that allow neurons to communicate. These vesicles release neurotransmitters into the synapse, the gap between neurons. To transmit signals, SV2A is a protein found on these synaptic vesicles, and SEL has to bind to it in order to work. This means SV2A is a potential target for developing new anti-epileptic drugs.
So SV2A is a lock on these neurotransmitter containers, and SEL is the key. This makes SV2A very important for designing new epilepsy drugs. One of the experiments involved incubating hippocampal slices with SEL, which led to a decrease in the relative amplitude of CA1 field excitatory synaptic potentials over a high-frequency stimulation train. This effect largely disappeared when spontaneous activity was removed by cutting the Schaefer collaterals during the loading period. Why is this finding significant?
This finding indicates that SEL specifically affects active neuronal circuits. When spontaneous activity is removed, the communication between neurons quiets down and SEL's effect is diminished. This suggests that SEL is most effective in the presence of ongoing neuronal activity, which is relevant to seizures.
That makes sense. They also conducted an experiment where they loaded SEL into hippocampal slices along with FM143, a dye used to visualize synaptic boutons. They noticed that slices loaded with both SEL and FM143 release the dye much more slowly than control slices during prolonged stimulation. What can we conclude from this observation?
This observation further suggests that SEL modifies synaptic transmission. The slower release of the dye could mean that SEL changes the dynamics of synaptic vesicles, possibly slowing down neurotransmitter release. All these findings suggest that SEL is acting like a brake pedal on overactive brain signals, which could explain why it helps with seizures.
We're seeing here that SEL seems to impact synaptic transmission in several ways, all of which could contribute to its anti-epileptic effects. It looks like this research could pave the way for new and more effective treatments. What else can we glean from these findings?
This study supports the idea that targeting specific proteins involved in neurotransmitter release, such as SV2A, is a promising avenue for developing more effective anti-epileptic medications.
What makes SV2A such an attractive target for new medications?
It appears to play a crucial role in regulating the release of neurotransmitters. By modulating its activity, we might be able to fine-tune neurotransmission and prevent the excessive neuronal firing that causes seizures.
This study specifically looked at SEL's effects on excitatory synapses in the hippocampus. Does this limit the applicability of these findings?
Not necessarily. While the hippocampus is a key brain region involved in learning and memory, seizures can originate in various parts of the brain. The basic mechanisms of synaptic transmission that SEL appears to affect are likely relevant in other brain regions as well.
So these findings could have implications for treating different types of epilepsy.
Potentially, yes. However, more research is needed to determine whether SEL's effects are consistent across different brain regions and seizure types.
What kind of research would be most helpful in building on these findings?
It would be valuable to investigate SEL's effects in other brain regions known to be involved in seizure activity. Additionally, exploring how SEL interacts with other anti-epileptic medications could be beneficial.
Are there any potential downsides or limitations to SEL that we should consider?
As with any medication, potential side effects need to be carefully evaluated. It's also important to remember that this study was conducted in vitro, meaning it was done using brain tissue slices in a laboratory setting. While these experiments provide valuable insights, They don't fully replicate the complexity of a living brain.
So further research, especially in vivo studies involving live animals or even human clinical trials, is crucial to confirm these findings and determine the safety and efficacy of SEL as a potential treatment for epilepsy.
Yes. It's also important to remember that epilepsy is a complex neurological disorder with various causes and subtypes. While SEL shows promise, it may not be effective for all forms of epilepsy. Personalized medicine, where treatments are tailored to the individual's specific type of epilepsy, and genetic makeup is a growing field that holds exciting possibilities for the future.
What would you say is the most significant unanswered question arising from this research?
I'd say the long-term effects of SEL. This study focused on short-term changes in synaptic transmission, but understanding how chronic exposure to SEL might alter neuronal function and potentially lead to adaptive changes in the brain is crucial.
So as we move forward, researchers will need to look into the long-term effects of SEL to determine its safety and efficacy as a potential treatment for epilepsy.
Absolutely. Investigating how SEL interacts with other anti-epileptic medications and exploring its potential in different types of epilepsy will also be essential.
It's clear that this research opens up some exciting avenues for future investigation. Thank you for guiding us through this study and sharing your insights. Now, wrap up our discussion, I want to remind everyone that today's episode is sponsored by Science.bio Premier Nootropics. You can find the link in the description. Is there anything else you think our listeners should keep in mind as we conclude our look at this study on Seletracetam? Now, to wrap up our discussion, I want to remind everyone that today's episode is sponsored by Science.bio Premier Nootropics. You can find the link in the description. Is there anything else you think our listeners should keep in mind as we conclude our look at this study on Seletracetam?
This research really highlights the complexity of synaptic transmission and the challenges in developing effective treatments for neurological disorders like epilepsy. While this study offers valuable insights into the potential of SEL and the importance of SV2A as a target for new treatments, there's still a lot we don't know.
It seems like there's a lot of potential here. But researchers need to keep digging to fully understand SEL's long-term effects and how it might interact with other treatments.
Yeah.
Well, thank you for walking us through this study and sharing your expertise.
You're welcome.
And to our listeners, thank you for joining us for this exploration of Seletracetam and its effects on synaptic transmission. We encourage you to keep learning and exploring. Until next time.